Namaskar, good afternoon, good evening, depending on wherever you are. Today it is my pleasure to welcome you all on behalf of the National Data Quality Forum, hosted at the ICMR National Institute of Medical Statistics, to the webinar series organized to discuss and disseminate information on measures being taken to ensure good quality data. My name is Dr. M. Vishnuvardhan Rao. I am the Director of ICMR National Institute of Medical Statistics, New Delhi. I will be moderating today's webinar on quality control and electronic data management of TB trial data. Clinical data management is a key step in clinical research, leading to generation of high quality, reliable, and statistically sound data, which further helps in patient management and policy making. It involves a series of steps ranging from proper collection of data to management of research and data as per regularity standards, that is complete and error-free, giving the reader confidence in conclusion of the research. Though large and big data sets are lucrative, unplanned management of such data can result in incorrect conclusions. Avoiding such situations is possible if one is aware of the common mistakes in advance and puts in place a contingency plan to good quality and proper data management upfront in clinical research, keeping in view the sustainable development goals of TB elimination by 2030, and the Honorable Prime Minister of India launched a TB-free India campaign and set the 2025 target for TB elimination in India. For this mega program, also we need a quality of data to assess and the progress periodically. REDCap is pleased to host a webinar titled Quality Control and Electronic Data Management of TB Trial Data. We aim to see the type of data quality issues that would occur and possible solutions for that. Before I invite Dr. C. Padma Priyadarshini to elaborate on this, it is my honor to introduce Dr. Padma Priyadarshini, my colleague and a good clinician and enthusiastic researcher, currently the director of ICMR National Institute of Research in Tuberculosis, which is a premier institution of ICMR. She has a short-term fellowship in HIV epidemiology from University of California, Los Angeles, a master's degree in clinical and translation research from Tufts University, Boston, USA, and PhD in pediatrics from Dr. MGR Medical University, Chennai. Over the last 20 years, she has been involved in multiple clinical studies and trials of HIV and TB among adults and children. She has more than 100 publications in peer-reviewed national and international journals and 4 book chapters to her credit. She has been involved in framing national guidelines for management of extrapulmonary TB, introduced by bedaquiline for drug resistance TB patients in India, and nutritional guidelines for TB patients. She was awarded the Lupin Toy Oration Award at the National TB Conference in 2016, Lucknow, and in 2020, Indore by the TB Association of India. Without further ado, over to you, Dr. Priyadarshini, for your presentation. Before she takes over, I have a small request and some set of rules what to follow. Ma'am, yours will be 25 to 30 minutes presentation, and again I thank you all for joining. As we start with just a couple of rules to ensure a smooth session without any guideline, any glitches. For this session, all participants shall be on mute except the speakers. We will be taking chat-based questions. The chat box shall remain live throughout the session. Please feel free to type in your question anytime during the session. Our esteemed speakers will respond to those towards the end of the session. Thank you very, very much. Over to you, ma'am.
Thank you, sir. Thank you very much. Okay, so I would also like to thank Dr. Ravindra Kumar Diwan, our director from NIPRD, who was joined and very graciously accepted to give his remarks and comments after the presentation. Thank you, sir. And thank you, our director from NIMS, Dr. Vishnu Rao, sir. He's been a friend and a guide for all our statistical input for the studies, and he's a very active participant of the TB Research Consortium too. Thank you, sir, for giving us this opportunity. If I can share my slides.
Yes.
So there's supposed to be a pop-up which did not come again. Can you see my slides?
Yes, yes, ma'am. Yes, ma'am. So please go ahead, ma'am.
I am not able to see my slides.
Okay.
This software doesn't allow you to see your slide, see it for yourself, but the audience is able to see it. So go ahead, ma'am.
But I need to see my slides for me to present it from my slides. So there is no way I can see the slides here, is it?
You will have to navigate through it, ma'am. If there is any other device, then you can put yourself on mute and use that.
Okay, then just give me another just 3 minutes for me to set up another device from where I can see this.
Just give you the presenter access one more time and we can see if that works.
Sorry for small technical glitch and we'll be starting.
Better?
It says show my screen, but it's not, I'm not able to see the slides on the screen. So I just set up another device so that I can see my slides.
If you can open another window, then that would probably solve the problem. I think if you see in your taskbar below, then I think you would have that option.
Okay, yeah. So thank you, sir. I can see my slides now. So very briefly over the next 20 minutes, I would like to discuss on quality control and electronic data management of TB trial data. So just in 2 lines for people who are not from the field of clinical research, it's basically medical research involving live subjects, live people. I mean, there are 2 kinds. One where you just observational studies, where you observe people in a normal setting. Researchers will kind of gather information on broad characteristics of these individuals who are involved in research and kind of compare any changes over time. Whereas the other group is the clinical trial. Clinical trial is what we are currently interested in. Okay.
Ma'am, any problem?
Yeah, my slides are not moving, so I was just wondering. Is slides moving on your side?
Yeah, clinical research is the title.
No, I've got the next slide of clinical trials. Did it move from your side?
No.
It's not moving, right?
Yes.
Slideshow mode and put it on it. No. We can no longer see it.
The team, are you able to restore it?
Ma'am has left the panel. We'll just make sure that she's back.
Dr. Rao.
Yes, sir.
Rao, any specific reason we are using this GoToWebinar platform rather than the usual Zoom platforms and other things?
There is no specific reason. Generally, we use Zoom here in NIMS. This is being organized by the Population Council, our partner. So probably they are using this. Actually, we use only Zoom. Very simple and easy.
Number of participants is not huge really, so that there should not be any difficulty with the simple platform.
Understand.
Because otherwise, if we have longer participant number is more than 500, then we are using this eco platform that we have.
Yes, absolutely, sir.
Maybe it's a better platform, but only thing is we are not very familiar with it.
Yeah, they have been doing it. I don't know. Did she join? Madam has joined.
Not yet, sir. We are trying to reach her out.
Is the net problem or she is?
I think it could be network issues on her end.
Correct. Kanika.
Sorry for the audience.
Should I call Dr. Padma and find out what is
Yeah,
happening?
yes, sir. Please do.
I'll do that.
Okay, people don't try.
Is it network issue or something? You are not visible at all. Hello. You are reconnecting. You are not neither visible nor audible, nothing. As if you are not joined it.
Yeah, I think ma'am is here.
Reconnect.
Ma'am is here. Ma'am is here.
Okay. We are waiting because nothing is visible. Okay. Dr. Padma says she's reconnecting. She was not even aware that she's not connected.
Can you hear me now?
We can hear you, but we can't see you. That's all. Okay.
Video has not been connected.
Yeah, it is off.
Acha, can you see me now?
Yes, ma'am. Very much. I'm very sorry. A technical
No,
glitch.
no problem. So can you then ask them to share the slides? I can
Yeah.
go without the slides because I think the problem was when I started sharing the slides.
I am making you the presenter again. I think now you will have access to share your screen.
Do that fast.
I would also request you to kindly share your slides with us. We can present it from our end.
So now are you able to see the slides?
Yes.
We are
Okay, ma'am.
able to
Please go ahead, sir.
Minimize this.
Oh, ma'am. Go ahead, ma'am. Go ahead, please.
And is it moving? See, this is what happens. I can't see the slides. If you,
You'll be able
Slides
to
are
see
not
your
moving
slides.
now, sir? Yeah,
Yeah,
can
No,
you
they're
see the
moving.
Yeah. Now
Now they are moving also, ma'am.
Okay, so from where do I start? Do I start from the beginning or should I start from, I don't know where I was cut off.
Yeah, third slide. Go back. Go up, please. Go one more up. One slide up.
Yes.
This is the first slide, sir.
First, second, just go to third slide, please.
This is my third slide.
Yeah, ma'am. Now from this slide onwards, the clinical trials.
Okay, so sorry for that. So as I said, clinical trial is one form of clinical research where you not only observe the patient but also do an intervention either in the form of medical, surgical, or behavioral intervention. And the primary researcher tries to do whether a new drug, diet, or medical device works in this participant. So as I say, there's a lot of data that is getting generated in these trials, which can be collected either in the form of paper, that is what we've been doing for years together, collecting on a case report form, either in the form of diaries or questionnaires. But now we have moved on to collecting electronically, electronic CRFs, by either using a handheld instruments in phones or tablets. The third method is where directly the end user can generate his data and put it onto the database. That is the direct data capture. That is usually done, for example, by the patient themselves when they use a pill box, pill box where they take out a pill and that directly gets entered into the database if the patient has taken his pills. The data collection also can be done by the investigator himself or the entire study staff. Now, why are these different systems of data capturing? As we know, the paper-based system, it's very inexpensive. You collect the data from the investigator or from the participant, but then you have to enter it onto database, which requires manpower. It also is time consuming. And when you suppose if there's a query generated, it again goes back to the site and they have to give you the answers to that. While the electronic data capturing, directly the data enters into the database, it is time saving. And if there is an audit or any electronic alert that comes out, it can be resolved immediately. However, it is expensive from the investigator's point. Now, all this data that is collected from either a clinical research or a clinical trial, either from human participants or from the laboratory results, and to meet mainly the objective of clinical trial, there's huge amount of data that's getting generated. All this is collected and that is known as database. As an investigator, I'm only worried about writing a proper protocol, doing a study, and then getting my results and getting published. So I should be worried about my study design and the analytic method or what is the outcome. But in between these two, there's a major step of the study conduct. Study conduct, one important point is how do you collect data? How do you manage this data? Because there should be proper, good quality data has to be collected. Missing data has to be avoided for me to get a good result. And therefore, any investigator, be it a clinical trial investigator or a clinical researcher, should worry about what is a database, where the data goes, what happens to it. Because in a study, you are supposed to be giving an interim result. The interim report not only is necessary to regulators like DCGI, but also for a data monitoring committee, for the scientific advisory committee, or even for the community advisory board, they might want to know the interim of any trial or any research that they're doing. Hence, your interim report is very important. Also to know how your study per se is going, the workflow, is it going properly? Are there any difficulties faced? What is happening to the data that is getting generated in midway? Does it need a midway corrections? All this can be, obtained if we have a proper data collection and a properly maintained database. For maintaining this database, there's something called clinical data management system. This is very important because this is what will give you good quality, reliable, statistically sound. The definition that Dr. Rao just said in the starting of this webinar, that is very important for a good maintenance of functioning of a clinical data management. And that gives you a lot of confidence when you present your data anywhere. If it is properly collected, integrated, and that is available at any given time, it is verified, validated, and then it all comes from a properly conducted clinical trial. Like we have good clinical practice, good laboratory practice, we also call something called a good data management practice. Now, this good data management practice will make us, if you kind of verify that we have a complete and accurate database, and whatever data that is collected is true, it is the integrity and quality of data that is being coming from a participant is true in the database system, sufficiently clean to support analysis. Sometimes what happens is you go on collecting data, you go on entering into the database without even checking what it is. And later you will find when you start doing the analysis, you will have, if you take a mean weight of this group, the mean weight might come out 1,344. Mean weight of individuals of age group of 30 to 60 years cannot be 1,344 because At some time point, you will see the weight that was entered. Instead of writing 31, it would have been 311. So all this has to be, cleaned and sufficiently good enough to have good statistical analysis, interpretable. Now, for this good data management system, where does it start? Whole thing starts from the time of protocol development, not at the time of doing the data analysis, but the protocol development. In the protocol, you have to have a data management plan, and the data management plan should be reflected in your CRF. If CRF should say what is the data you're collecting. Dr. Rao, am I still on the screen, audible and visible?
Yes, ma'am. Data management process you are in and is audible and visible.
Yes, sir. Thank you, sir. Therefore, once you've designed a good CRF, whether it is an e-CRF or a paper CRF, you should also simultaneously design your database. What is going to be entered, who will review or validate, how will you resolve the discrepancy coming out of it, and finally, you should know when you want to lock your database for analysis. And the last point would be archiving all your data that is generated and stored data. For all this, there are two important mechanisms: quality assurance and quality control. Now, this flowchart tells you how the data is getting generated. What is the procedure? You start writing your CRF, then you circulate it among your peer group, you get comments from them, redo your data collection form, then you finally freeze your data collection form. Once it is frozen, take it to your data entry person or data manager and the statistician, sit with them, see whether that will answer your protocol objectives and outcome. They also will give you comments. Again, you freeze it. So please freeze your paper CRF or the CRF before you ask your data manager to make a database. So once the database is done, it is very difficult for them to go back and change the database to keep on adding or deleting entries. So that is what it says. Once its correction is all done, then complete the data corrections, go to them for making a database, but then also put in the quality check and the quality control mechanism. As I said, data quality is very, very important. It is a greatest asset to you. You have a good data quality. As I told you, as an investigator, you feel more confident to go on stage and present your data to any number of audience. At the same time, it is your credibility that is also a question here. By credibility, of course, you publish your data, you present your data, that is fine. But what I mean by credibility here is, if you present a wrong data somewhere, one time everybody will say, okay, a mistake was made. But if that continues, people will start questioning the way you collected the data. Maybe you're giving all falsified data, fabricated data. So your credibility as researcher itself will come to a question if your quality is not good. Hence, make sure it is C3 or C cube T. It should be correct and accurate. It should represent exactly what you got from the field. Consistency of the data is very important. As an today, I made a weight of 35 kg and 2 months down the line, I write 350. So the consistency of the data is very important. Make sure the data is complete. The missing data has to be minimal. In a live scenario, there will be missing data. I won't say this 100% data will be available. There should be missing data. If you see 100% data, again, that's credibility. There's a question mark there. And timeliness, that is very, very important in clinical research. Right data to the right person at right time is important. Otherwise, we all miss the boat. So it's very important for clinical researchers. So when you maintain the quality of data, make sure your data is complete. It is clean. You have to have some algorithm or rules by which you can make sure there is no duplication of data. Sometimes what happens, everybody starts doing data entry and you find the same entry being written 3 times or 4 times. And that will kind of add up or add on to your number of patients enrolled. So this the duplication has to be avoided. Merge any duplication of data that's available. So all this should be coming up in your SOPs, and make sure there is a rules for security also. The data quality management is very important. There are only 4 pillars: quality planning, quality control, quality assurance, and finally it's a quality improvement. Quality planning is have a plan, have an SOP of what will be collected, who will check it, how is it validated. Quality control is nothing but monitoring, real-time monitoring. As and when I keep collecting the data, somebody checks it, somebody monitors this. That is my quality control. And make sure that this quality assurance is there. This is a planned systematic activity. Here I assure the regulators this will be monitoring will happen on a day-to-day basis. The quality assurance will come with a systematic plan. Once 6 months is completed, or once I've enrolled like 90 patients, something like that. So put in a quality assurance plan, systematic way of checking your data. Finally, bring in a component of quality improvement. Now the term QA and QC kind of they use interchangeably, right? So quality assurance and quality control. Briefly, what is QA and QC? QA is a process, it is a continuous process. You make sure all your activity that you're doing, it is a process that is being measured. Your quality control talks about the product. If you see, these are the definition that has come from the industry. By mean even drug industries or device industries, but very well fits into clinical research or clinical trial. For quality assurance is a proactively the investigator is making sure the quality of his work is being done, whereas quality control is a reactive or knee-jerk phenomenon to quality assurance. The quality assurance will improve. It's an ongoing process. It's a daily process. Anybody in the team can get involved in this. It has to be verified. Verification is one important component of a quality assurance. Whereas a quality control will make sure that errors are being picked up. You identify specific person to do the QC of your protocol. It is more of a validation. I give you a result. Somebody comes up and checks whether the result that I've got and the methodology I have followed to get this result is correct. There are no errors, and that person is validating my result. So that is the difference between QA and QC. QA is a process. You go on. It's an ongoing process. Whereas quality control is a correction. It's a corrective tool. It is for the end product. So this is what talks about quality assurance. Now, quality control can happen in two ways. There's something, again, a new term, monitoring and auditing should happen. Now, monitoring, again, the whole team, the investigators team can do the monitoring. It could be a person who is not involved in that protocol, but he is part of an overall team. For example, if NIRT is doing a multi-centric study, I will make sure nobody in my particular study team does the monitoring, but there will be somebody from NIRT's other department or NIRT's whole overall team will go do the monitoring of sites. Whereas auditing is done by totally a third party. It is either appointed by the sponsors or DCGI can send them somebody for auditing. So auditing is more, it's a regulatory thing. Monitoring is a part of good data management policy or good clinical practice. Every study should have a monitor who is not part of the study but can be the part of the overall team. And if you do go on making lots of mistakes, you do a lot of protocol deviations and violations, you will attract auditing, and that is when it becomes a part of the DCGI processes or the FDA process. So where do I start talking about this data quality? You have to start at the time you start writing a protocol. Once you fix up your primary and secondary objective, you should have a pre-planned analytic plan to get the outcome. So it should go into the protocol and make sure whatever objective that you want should be reflected in your CRF or the data collection tool, which is a case report form. This case report form, as I said, could be a paper copy or it could be an electronic copy. So one important take-home message would be, what is the thing that we're collecting? Sometimes the investigators are so enthusiastic, they want all kinds of data onto the study. They kind of lose track of what was their objective and what is the outcome. So please collect only data that will help you answer your primary and secondary objective. Avoid collecting redundant data. And if you have a statistical plan in place, that will help you to tell what kind of a data you want. Sometimes, for example, I can give you in TB trials, we would have written there time to sputum culture conversion as per my secondary objective. But then when I start collecting data in my CRF, I would have left out one particular place where I should be collecting time, days or hours. That particular boxes, or it will not be there in the CRF at all, which I may not realize it. At the end of it, when I do the statistical analysis, well, then I know I cannot answer my secondary objective because no data was collected on time for conversion for individual patients. That's why if you have the analytic plan, whenever you have the analytic plan, make sure your objective and outcome points are met. That is why you have to adequately review your CRF before they're finalized and they go for e-data entry or for creating the database. Always keep your end user in mind. So as an investigator, I want to collect 101 things. I make a lovely CRF, which the end user will spend almost 3 hours with the patient to collect the data. An end user may be a field investigator who has just passed his +2 or +10 or a basic qualification. He gets bored collecting your data. He doesn't understand half the jargon in my CRF. The patient becomes bored answering your question. So always keep the end user and see where the source data is. As much as possible, avoid giving them free text boxes. Collecting so much of free and written notes, which may not even be used later. Preferable to use yes or no check boxes. There are 5 principles to know the data that you have collected is good quality. It has to be accurate, legible, complete, original, and attributed to who actually collected the data. These are the 5 points. Now, accurate. Now, the trial data should capture exactly the source data. Any corrections or any changes that you have done in your document has to be signed and dated. This is one important criteria for any clinical trial data that you collect, audit trail, leave an audit trail. The example on the right shows someone has done a correction on between neurological, normal and abnormal, but they have gone ahead and signed and dated it when they have made this correction. That gives you more confidence. Whoever has corrected it has owned up the responsibility. That makes it more accurate. Legible. So this is an example of a good legible handwriting in one of the CRFs. Do not overwrite, scratch, whiteness. Don't do all that. Clear handwriting on CRF is equally important. And whatever you do, it should be readable. I know doctors' handwriting are the worst handwriting, but that is why you keep it more of check boxes and ticks instead of free writing. The data should be complete. Suppose if I say this is a chest X-ray that had to be taken, and maybe at this visit there was no chest X-ray and it was not taken. In that case, you're right. So you did not mark S, but then you have to mark no. You cannot leave it blank. Here I have a form where X-ray was not done because it was not asked for this particular visit, but nothing is marked in this form. So this is not complete. Now I have to go and think whether it is not applicable or it was not done or patient went away. So this is avoid blanks in a CRF. Data must be recorded at the time the activity occurred. Not now. It's not like now the OPD is so busy, I was not able to fill. I'll remember it later. So after 3 o'clock, I'll fill my CRF. That's something wrong. So your data form has to be filled at the same time. And the fourth principle is original, the accurate transcription of source data onto the database. Here the example on my left side, I'm showing you a source data where if you look at the second column, it says one of the drug DLM is sensitive. But when that was transcribed onto the database, it has become resistant. So these things, when somebody reads it without the source document, they will read only the original data that is entered onto your database. It will be marked as resistant and that will increase your, it will falsify your analysis. So this has to be very, very important. Your data has to match. Accurate transcription of the source data is very important. And the last principle being attributable. So quickly, the 5 principles are accurate, legible entry, complete entry, original or the accurate transcription of the source data to the database. Then last important point principle is attributable. Who has done who has recorded this information? So have a designated study staff to record it and have somebody verify it. Always leave audit trail, make them sign and date your data. This slide about its data collection, data quality does not apply only to clinical trials or clinical research. Even if it is program data, even if it's epidemiological data, the lab data which comes out, all this should have a proper data quality. The data quality has to be checked and monitored. Make sure your quality assurance is there for the lab. It is also there for the program data or the field data. So that is about why data quality is important. How do we collect it? Very few words about the data capture, electronic data capture that we follow here at NIRT for all our TB trials is something called REDCap is what we use here. Here what we do is from the time the protocol is designed, simultaneously we start working on the CRF, the data collection tool. And once we circulate it to all our colleagues, everyone gives in the input and everyone, half the time we don't agree with each other. And once when finally it is done, we go to our data managers for creating this database. And even if the database is created, we are not happy. Again, someone gives a last minute entry, the database has to be changed. And our electronic data monitoring people are extremely smart. They know they will never, even if the database is ready, they do not immediately give it to us for use. They want to validate our database. They will only open a pilot version, make us do all kinds of corrections on that before it is locked and released. Once it is done and all our multi-centric study people have been trained, database entry happens. Wherever there is a discrepancy, that is reconciled. And finally, when we're ready to do the database data cleaning, after that, database is locked before we take it for the final analysis and report writing. REDCap is nothing but research electronic data capture system, a secure web-based application for building and managing online data forms. Now, though this was developed in 2004 in Vanderbilt University, we still have been using this form for many of our studies. And I know NIMS is coming up with our own data capture tool, and that is our ultimate aim, to have an India-based or ICMR-based data capture tool for TB trials and TB research work, so that one single forms can be used across the country for capturing of TB data, and we can have a mega database for the TB trials or TB studies. So when you design any database for capturing of TB trial data, it should be easy to use. Fast and flexible is what we need. If there is a correction that has to be made midway, we should be able to do it without crashing the entire database or data that was collected earlier. It should be secure, stay within our domain itself, not move out to some other cloud or some other access. Mid-study modification should be feasible, and we should be able to easily export our data, because our statisticians, who are also the end users, they have a specific package that they're very well versed in, like either SAS or SPSS or R. We should be able to export the data that is got from these databases. Now, the REDCap, why we have been using, because it gives us an easy access to any kind of field that I want. It gives me both free text writing, unstructured field type is also there. I can also give access to the clinicians to write clean clinical notes, survey notes. At the same time, we also have structured field types where you have multiple answers to a question, or the drop-down menu, or a radio button. All that is available. So both kind of writing can be done in this REDCap database. It also giving me an access to upload the photos and X-rays. Like, all the TB studies, you do generate a lot of X-rays. These X-rays can be uploaded. In fact, it also has a feature to upload your consent documents. So these would be something which we will be looking forward to if we are going to develop our own database for data collection. REDCap also comes with an excellent resource videos, or it tells you what and all you can do and what cannot be done. Now, the data manager should also have an access to a dashboard. This dashboard will tell you if a patient has come to 3rd week of follow-up. I can always go back and look whether the 1st week and 2nd week data entry has been done or not done. In sitting here in Chennai, I'll be able to monitor this across the sites. If it is not done, all we have to do is just call them, ask them to complete and do the data entry. So this kind of dashboard access is very, very important, whether it be clinical trial or it's a survey or the surveillance project, if it is being done on a multi-centric sites. We can also build our CRF on to this database. And these case report form, as it's saying, it is for TB trials. So it's very standard questions. It will be cough, the duration of cough, whether it is hemoptysis, breathlessness. It gives you both free writing as well as radio buttons. You can choose yes or no kind of an option and give the days in weeks or months or whatever. We try to avoid as much as possible freehand writing, but even then we can have text boxes, leave some notes for the doctors to write. Earlier, we used to fill the paper form where you have blank sheets where the doctors can write the notes. Here, it gives you free writing. So when you're transcribing from the CRF onto this database, our data entry operators can put in the notes in this box the doctors have written in the source document. The radio button options also available, and we also have multiple checkboxes. Suppose if I have multiple times my patient has taken TB treatment, it gives you an option of any number of times I can write where did he take treatment, what treatment was given, how long was given. So all this is needed for the electronic database that has to be done. And if there is ongoing activity, for example, there's an adverse event. Today when I fill it, the patient is still ongoing, but probably 4 months later it will get resolved. I should be able to come back to this form, pull out this form and write their result. So these navigabilities across the fields and across the pages should be given to any new database that we want to create for TB trials. So we have been using REDCap now for many of our trials, and we were able to create all these kinds of forms on the REDCap, and that is what gave us an easy accessibility to do multi-centric studies, where we are also able to upload images, upload CT scans, upload chest X-rays. Not only that, when you do a behavioral study, it can also help you mark something which is a qualitative answers. For example, if I say how mobile I am, it can say from 10 to 100 is my scale. It's able to mark what is the scale of your mobility also. Suppose if X-ray is unclear, you can always delete the X-ray and uploading different kinds of X-rays. Now for the current 2 studies, yes, it's at a couple of last slides. We're also able to randomize patients on REDCap. And once somebody is randomized, we should be able to create a calendar schedule for the patient, saying that when he should come for what kind of follow-up. If there are queries that is raised from Chennai, the queries easily go online through the REDCap database itself to other sites, and we're able to make the correction of the mistake. For example, if you look at this, the previous weight here was 65 kg, but with mistake, the data entry person has written here as 36 kgs. This was immediately flagged by the database itself, and they were able to correct it before they went on. So far, we are able to raise all these queries. 95% of them have been resolved. We should also have a data quality module, data export mechanism in such kinds of studies. So whatever I say, I want to end with the saying that any number of database, any number of eCRF, electronic work, it is all good and nice, but nothing like a human touch, nothing like having your data entry people, your data managers all sit in the same room, talk across the table, do the corrections, take the inputs and correct it. So still face-to-face meeting is more welcome than any electronic meetings. Thank you.
Thank you. Thank you very much, ma'am. That is excellent, very detailed presentation. Now I move on to it is also my pleasure to introduce distinguished discussant of the webinar, Professor Dr. Ravindra Kumar Diwanji. Professor Diwan is the director of the National Institute of Tuberculosis and Respiratory Disease. He is a senior thoracic surgeon in the country and has been a pillar of thoracic surgery not only for the institute but also entire North India. He has developed a high-quality thoracic surgery unit at the institute, which is engaged in the most complex thoracic surgical restrictions with outcomes comparable to international standards. He is always strived for academic excellence with more than 30 publications of his work. He is the founder member and the vice president of the society and is actively engaged in promotion of thoracic surgery expertise globally. He is a member of several professional bodies, including European Society of Thoracic Surgeons, Indian Association of Cardiovascular Thoracic Surgeons, Asian Society of Thoracic and Cardiovascular Surgery, National College of Chest Physicians, Indian Association of Bronchology and Lung Cancer Society of India. I now invite Professor Diwan to provide his observations and remarks on the topic and share his experience as an expert in this particular area. Sir, over to you.
Thank you so much, Professor Rao. Dr. Rao, and as all of us have seen, that Dr. Padma Priya has given us very succinct and elaborate analysis of the issues regarding data management, its capture, and all the difficulties that we encounter. Most of the research which is being carried out in the field of tuberculosis under the guidance of ICMR these days is being generally guided by the NIRT, National Institute of Research and TB, under Dr. Padma Priya's guidance, and our institution being one of the institute generally associated with most of the clinical trials. We are dealing with day-to-day difficulties that are being observed, and I think I would like to flag certain issues. Most of her talk is very extensive, elaborate, as well detailed about the issues. I think about all the data is so important, because whatever we are trying in terms of researches as well as planning and whatever programs that we are making for TB control, they are based on whatever recommendations we people are making, and it should be based on an accurate scientific research. So data authenticity is the most important issues. And the issue that I wish to, which I think Dr. Padma Priya has also raised, that when the data is being collected, are we being lethargic about it? I think that is the most important point. When we are planning about it, who is collecting the data, whoever he or she is actually feeding absolutely correct data. And I have seen that, as she has also mentioned, that sometimes mistakes are seen. And then integrity, whether honesty and dishonesty, and all of us, what is my experience throughout, that whenever we train our people specifically that you're doing a research, so the entry of data has to be accurate. I think in our residency programs as doctors, sometimes because you have to fill up, say, 100 case sheets in a day, sometimes we are in a rush and we are not accurately filling in data. The same trend cannot be continued when you are filling up a research data sheet, and errors inevitably creep in. And once an error creeps in, now most of us may be knowing that, say, from 1992 onwards, our TB mortality has gone down by 42% by 2015, etc. But sometimes we feel, has it really gone down? It has not, because our clinical experience says almost the same mortality exists. So has our data capture been absolutely correct or not? That has to be seen. That's the most important point. I think I would, after my discussion, I would invite comments from Dr. Padma also, whether she agrees with it or not, that our data at the initial site of capture is most important. Whether clinical data, whether data for clinical research, whether data for epidemiological research, or whether data for basic research, whatever it is. So planning at the beginning of the trials itself is very important. And because she is involved, and most of us are involved in multi-centric trials. Now, situation in Bombay, NIRT may be quite different from situation in NITRD or Agra. So I think a combined meeting of all the sites is very important. At that time, we can discuss many issues, how data will be collected, because there may be specific situational difficulties in data collection. And sometimes that gets overlooked if there's a centralized command of designing a research trial and then putting it into operation. Because there will be, because I know the data is being collected, suppose in MSF in a Shivri center, which is just a primary health center, or it is being collected at National Institute of TB and Respiratory Diseases. The quality and its accuracy and various situations will vary. So planning at the time of trial is most important. I think if we spend 1 month extra in planning a trial is worth it rather than going ahead with the trial. And related to this, the question which comes to my mind, all of us in various conferences speak about in the context of TB, that our Prime Minister has given a clarion call to eliminate TB by 25. So that puts a kind of pressure. I think that pressure should not force us to design our trials poorly or to hurriedly give clearance to situations. That is something which, as medical professionals, it is our duty that we see to it that this does not happen. We apply our mind completely and do the planning in a complete manner. Sometimes it may lead to inevitable delays, and I hope the participants do not include. As medical professionals, we do know that certain things have constraints, and we have to do, if we want to do be honest to ourselves and true to ourselves, then the data collection has to be, it cannot be really bound by time limits, though we would like. And second, but she mentioned that we have to do in a proper time limits have to be maintained. For instance, if you've seen a patient today, that patient's data has to be entered today. You cannot wait for 2 weeks. And third is clarity about the methodology and purpose of the trial. I think that has to discuss with everybody. And now that everybody does not include only the principal researchers from various sites. That may also include the people who will be actually collecting data. And sometimes discussion with the people on the ground, like data entry operators or the field investigators and the principal investigators, are much more important in the beginning, how we will be collecting data. About data cleaning, she mentioned that duplicate data, the same data about the same patient can be entered repeatedly by several people. So that data cleaning, we have to be, I think she has also mentioned, repeatedly go into data. I think every month has any inadvertent error being made in the data collection. Some duplicate entries have to be done, and inadvertent errors do creep in, and they have to be cleaned. But data cleaning in a given site can actually degenerate into data management. I mean, whatever is an inconvenient finding, I will omit that, and whatever is convenient finding, that will be continued. So that should not happen. So she has mentioned about data monitoring. I'm very happy to note that she has mentioned the data monitoring team is different from those who are generating data, and those who are auditing the data is an absolutely different team. And but data monitoring should not be done only at the end. It has to be done in between. And I think at the beginning of the protocol itself, we can see that how many times we will monitor and evaluate the data, so that we can do our data cleaning periodically rather than doing just at the end. And I think it has to be done at various levels. Various sites have to be and then the central site has to do it. And within the site, I think various small local groups have to do it. Maybe our team. So for instance, we are doing the DeepL trial. Now our team here actually sees that all the data every week, so that we are correctly doing the data. Then at the NITRD site, we can do it every month. And maybe with Dr. Padma, we can do it every 2 months or 3 months, whatever is necessary, whatever is decided at the beginning. So, and then data confidentiality. Despite we know that because we are transferring at various platforms, there is a risk of data getting leaked, especially the patient data. Of course, there are safeguards, but we have to look into it that data confidentiality is not compromised. Now, another issue, because TB data is also being entered into Nikshay portal. Nikshay portal has certain issues all of us have discussed in various conferences. Those issues need to be, and those issues, they are getting resolved and they need to be resolved fast. Another issue I wish to flag was about, say, difficulty in conducting some clinical trials. For instance, I am a thoracic surgeon. I wish to have absolute clinical trials for thoracic surgery. Now, they are extremely difficult because suppose we see a localized cavity. We say, oh, it's obviously justified to remove this cavity. But does it actually lead to better results in terms of long-term management of a TB patient or not? We do not have the data, and most of the data of thoracic surgery is based upon expert opinion, retrospective analysis, or case reports. There are generally hardly any prospective trials and are very difficult to actually design also. And I think I have organized a meet next week only, in which we'll be hearing Dr. Padma's ideas about how to design data and how to design trials in this field. So that will be an important area. And the point that she mentioned, that the data has to be accurate, legible, complete, original, and attributable. I think this is the point. This point I have noted with a lot of interest, that the data has to be attributable at every point to whoever has generated the data. About the use of the various softwares which allow us to transfer X-rays and CT scans, etc. I think we are still having problems even at local levels. So I don't know if it is, are we technologically savvy enough to be sharing it at all the platforms? That is something which maybe you can enlighten us better later on. Is it possible? And I think these are the issues I wish to flag as the team manager of the team which carries out. We have 33 to 34 projects going on in NIT RD as of now, and it does become a challenge occasionally to be able to periodically review them. Are they going along in the correct form? And the data is the essence of everything, because whatever analytic methods we have. And last point, which I suppose most of, being myself being a surgeon, sometimes the statistical tools being used to analyze data are becoming far too complex for medical professionals to even understand. And then we cannot just keep on giving it to statistics. Ultimately, the person who is the principal investigator researcher has the responsibility to understand everything about it. So we have to somehow design an element of simplicity to whatever analytical methods we are using. So these were my initial thoughts as the person, in the position that I am. And I would, of course, invite comments of Dr. Padma and Dr. Vishnu also. Thank you. Thank you so much.
Ma'am, do you have any comments to make, madam?
Sir, thank you, Dr. Divan, sir. Sir has listened to the talk very closely. Thank you, sir. He's kind of summarized my entire talk in a couple of minutes. And Alcov, I totally agree, sir. Accurate, legible, complete, original, and attributable is something must. And I agree, the whole business of clinical trial is expensive. That is why it is expensive, because everything depends on the data generation. So if you see all the clinical trials or clinical studies that we do, one place that we never compromise is getting a data entry operator exclusively for this particular study. Even the previous studies that we have done multi-centric, we have made sure whether there is, even if there's no medical officer, we want definitely a data entry operator. Because the medical officer would be called for 101 things, go to the OPD, go do that, or the lab person. But data entry operator's only job is to make sure there is no missing data, it is properly transcribed, it goes to the database and it's properly transmitted, queries are answered. So no compromise on the quality of data, that has been a mandate for NIRT. As far as sharing of X-rays, sir, yes, it has been a challenge in many databases that we have also found, because even for the TB vaccine trial, the HICON Rift, there was another database where also there was challenge in uploading the X-rays and CT scan. But as you know, sir, TB, you cannot do anything without chest X-rays. So that is why we still stuck on to this particular database that we are now using. And we are able to successfully transfer or transmit X-rays from other sites to NIRT. So the request would be from Dr. Rao is, we should have our own database, sir, our own Indianized database for various studies. You know, for TB, we need CT scan and chest X-rays. That has to come up. And that is what we're trying now. And we definitely would get involved with NIMS also for the coming up with ICMR specific database or data capture center, where we will be able to upload X-ray. For example, the next week we're having the surgical meet on the role of surgery in TB. There'll be so many films, so many short films, so many slides that they would want to upload if they want to make a database on surgical surgery and TB. We need to provide a data platform for that. Currently, we do not have, everything has got a capacity. Only this much of X-rays can be uploaded. But for when it comes to surgical input, there'll be more. I anticipate slides from pathology. I anticipate short films of surgery itself happening, all that. So we'll have to figure out, and we'll definitely come out with something for that, sir.
Okay. So thank you very much, ma'am, and nice presentation at the same time. Nice summing up of what you have told, reiterated, Dr. Divan sir. So I also take this opportunity Actually, you have covered all the 3 stages of where the possibility of data quality errors would come, like before the survey, during the survey, and after the survey or after the experiment. So that really helps us. Ma'am, we have one or two couple of questions to you, small questions. Would like to I'll just ask you these questions by one Mr. Abhimanyu. In the first step of the process, how is a protocol different from the research proposal and a concept note? His question is, in the first step of process, before CRF instrument, how is protocol is different from research proposal and a concept note?
Okay, so the concept note is some idea that I get as a researcher. I put a very brief concept note of what my objective would be, what would be my outcome, and how do I want to do the study design. It's very brief. It is not in detail. Whereas when you write your research proposal or your protocol, your research proposal should have everything in detail. How will I, suppose if I say I do a prospective study, my concept note can be simply just a prospective study. But when I write my proposal or protocol, I write in detail of where will I go for my patient enrollment, who will do the talking to the patient, who will fill the CRF, who will collect what data, how will it be brought, and what are the investigations to be done, what is a normal value, what is considered abnormal, what is inclusion, exclusion, in detail. So that's the basic difference between a concept note, which can be very brief, just a general outline of objective, outcome, methodology, timelines, and if you have a budget. Whereas your proposal should be in detail of everything, of how you do, what you do, why you do.
Okay. So I have 2 questions on REDCap, but I merge them into one and ask you. In REDCap, do we have the option to capture geographical location? Also, whether the random allocation of the patients is being done in the REDCap itself, software itself.
Yeah, so REDCap is, yes, you can do randomization on REDCap. It all depends on how well you are well versed with REDCap, because the certain REDCap doesn't have stratification. So you cannot stratify, but you can definitely randomize on REDCap, which we are currently doing in one of our studies. Stratification is also there, but we need another module to be added on to it. So the more and more you use REDCap, you become well versed with that. And REDCap also has a way to catch the geography. The GIS can also be used on REDCap.
That's all. I think there are no questions, Kanika. That's all. So thank you very much, sir. Madam, for your nice presentation. At the same time, Dr. Divanji for great elaboration of the presentation, and also the participants who are all present here and actually watch it this very keenly. This is very important topic. In fact, now the data is the new oil. If the data is not in all sense a perfect data, a data which is error free, then whatever the conclusions we are making will have a lot of repercussions on the policy. So now the era of big data, and we also should be careful about the quality of the data. Thank you very much, ma'am, for accepting our invitation and delivering an excellent talk. And Dr. Divanji, thank you very much for sparing your valuable time. I know how busy you are, a thoracic surgeon, I understand. Thank you very much, sir. So thanks for the team. Thanks for the team who has conducted this, though there are some glitches, and next time we will take care of those. Thank you very much, and thanks one and all. Namaskar.
Thank you, sir. Thank you all.
It's a very enriching talk, and thank you so much, Dr. Padmapriya. I'm also enlightened further. Thank you so much for giving me an opportunity to be part.
Thank you, sir. Thank you.